CAR T-cell therapy is a form of therapy that involves genetically editing the human T-cell. This edit inserts the gene that codes for the production of the CAR protein, or the chimeric antigen receptor protein. Adding this protein allows the T-cell to seek out and kill otherwise undetectable cancer cells. In acute lymphoblastic leukemia, the CAR protein is coded to seek out the CD19 molecule. This molecule is found on the surface of cancerous leukemia cells. Once the chimeric antigen receptor that is located on the surface of the T-cell finds the CD19 molecule, it bonds to it. Once the receptor and the molecule have bonded, the chimeric antigen receptor sends a signal to the T-cell. This signal activates the T-cell and causes it to kill the cancer cell. The CAR protein can be coded in many different ways in order to locate different molecules, but the particular molecule that is found on acute lymphoblastic leukemia cells is the CD19 molecule. So how does CAR T-cell therapy actually happen? First, blood is drawn from the patient's body using an IV. Then, the white blood cells and T-cells are separated from the rest of the patient's blood using an apheresis machine. Then, the patient's T-cells are sent to a lab to be modified, and the rest of the patient's blood is put back into the patient's body. In the lab, a virus is used to insert the gene that codes for the chimeric antigen receptor into the T-cells. This causes the receptors to be produced on the surface of the T-cells. Then, the newly modified T-cells are put back into the patient's bloodstream. Some chemotherapy may be applied before in order to kill some cancer cells and create space for the new T-cells. Once the new cells are back in the patient's bloodstreams, they are attracted to the CD19 molecule on the cancer cell surfaces. Once the CAR T-cells find the cancer cells, they identify them as cancer cells using the CD19 antigen, and then destroy the cancer cell. That is how CAR T-cells track and kill cancerous cells.